This is Masashi Ishikawa from the Department of Anesthesiology and Pain Medicine at Nippon Medical School. In this presentation, I'd like to talk about our research into the various effects of anesthetics via microRNAs. The main purposes of general anesthesia are sedation and analgesia that ensures safety during an operation. Its use is based mainly on experience and the results of clinical studies. Few studies have actually looked at the molecular genetic mechanisms involved in general anesthesia, and many aspects of these mechanisms are still unknown. In addition to their sedative effects during surgery, anesthetics also affect various organs and disease processes. They protect the heart and kidneys against ischemia reperfusion injury. They protect against lung injury due to inflammatory disease. And they can show either anti-tumor or tumor-promoting effects in cancer cells. In a previous study, we performed the first comprehensive measurements of in vivo genetic alterations caused by Sevoflurane anesthesia. The heat map in this slide shows the results of microarray analysis in various organs. We found that inhalational anesthetics affected gene expression in 1.5% of 10,000 genes. There were three main effects. 1. Inhibition of diurnal variations in gene expression in the brain. 2. Effects on the expression of genes related to drug metabolism in the liver. And 3. Expression of genes that regulate vascular tone in the pulmonary circuit. Based on these results, we used reverse transcription polymerase chain reaction to perform detailed examinations of changes in gene expression under exposure to various anesthetics. The results showed increased expression of genes related to drug metabolism in the liver, and the pattern of gene expression differed between intravenous and inhalational anesthetics. Genes related to the constriction and relaxation of the pulmonary vasculature were expressed simultaneously with inhalation anesthetics. Also, with inhalational anesthetics, we found alterations in circadian rhythm via changes in expression of circadian genes. Next, we examined the molecular biological effects of general anesthesia by utilizing proteomics and metabolomics as well as genomics. But we didn't find any chain of association of interactions between genes and gene expression to protein expression and metabolite variations. So we hypothesized that the expression of microRNA involved in the regulation of gene expression plays a role in this process and examined microRNA expression under general anesthesia. MicroRNAs are single-stranded non-coding RNA molecules with 20 to 25 nucleotides that function in post-transcriptional gene regulation of mRNA via mRNA degradation and translational repression. When some stimuli, like anesthetic agents, enter the cell nucleus, precursor microRNA is formed from nuclear DNA by Drosha cleavage. Precursor microRNA is cleaved by dicer transactivation response element RNA binding protein complex and becomes a mature microRNA duplex. MicroRNA-induced silencing complex, or RISC, is formed by argonaute or AGO binding to mature microRNA. This can move out of the cells via microvesicles or exocytosis in endosomes. MicroRNA-induced silencing complex inhibits expression of its target mRNA in three ways, depending on the sequence complementarity to the target mRNA sequence. In full complementarity, mRNA cleavage with subsequent DNA degradation occurs. Or, in partial complementarity, transcriptional repression or deadenylation occurs. The target protein expressions decrease due to mRNA inhibition by microRNA-induced silencing complex, leading to cell activity suppression. There are more than 1,500 microRNAs in humans, but their roles in normal and pathological cellular function are still unknown. MicroRNAs modulate various aspects of cell biology, including cell differentiation, proliferation, apoptosis, embryonic development, stress response, stem cell renewal, and metabolism. We therefore attempted to elucidate the mechanisms of action of anesthetics based on changes in microRNA expression, which affect mRNA expression. We comprehensively measured changes in microRNA expression. Some microRNA profiling studies have shown that both Sevoflurane and Propofol affect microRNA expressions in the liver, lung, and brain, and reported changes of 50% to 80% under anesthesia. There is a specific pattern of expression after each type of anesthetic exposure in each organ. However, the pattern is not altered by the anesthetic concentration. For example, of 177 expressed microRNAs in mice liver, 46 microRNA expressions were changed after Sevoflurane or Propofol exposure. In particular, there was significant difference in the expression of MIR1423P, MIR29A, and MIR378 after Sevoflurane and Propofol exposure. There may be specific events among the anesthetics that induce unique changes in microRNA expression patterns in organs. Therefore, post-transcriptional factors such as microRNAs that may control the regulation of gene expression are expected to play a crucial role in the biological effects of anesthetics. Other studies have also shown that general anesthesia exerts a variety of effects, such as organ protection, tumor promotion, and tumor suppression, by altering microRNA expression. The topics that we examined were whether the protective effects of anesthesia against organ dysfunction are mediated by changes in microRNA expression, and how expressions change in specific diseases. MicroRNA-mediated effects of anesthetics have been described, including protective effects in ischemia reperfusion injury and in lung injury secondary to septicemia. Sevoflurane exerts hepatoprotective effects by inducing MIR1, 17, 133, and 205 expressions in ischemia reperfusion injury. These target the AKT glycogen synthase kinase 3 beta, or GSK3 beta, cyclin D1 pathway. Sevoflurane promotes the PTEN phosphoinositide 3 kinase AKT signaling pathway by MIR17-3P and MIR27A expression changes, resulting in renoprotection. Sevoflurane also ameliorates systemic inflammation in the acute lung injury model through MIR155 downregulation. These findings gave rise to questions about microRNA expression in diseases such as cancer, and then to the concept for the present study. Examining the effects of anesthetics on cancer and other diseases may lead to improvements in the long-term prognosis and in the safety of perioperative management. Inhalational anesthetics modulate microRNA expressions, leading to cancer biology changes. Isoflurane exposure to ovarian cancer cells increases glycolysis via the PI3K AKT pathway and HIF-1 alpha by MIR21 upregulation as an oncogene. Sevoflurane inhibits cancer cell proliferation via PI3K AKT pathway with MIR203 upregulation in breast cancer cells. In colorectal cancer, Sevoflurane reduces cancer invasion and migration via suppression of the ERK pathway and MMP9 by MIR203 upregulation. And in glioma cells, Sevoflurane increases MIR637 expression, resulting in decreased cancer migration and invasion via AKT pathway inhibition. Propofol exposure alters anti-cancer microRNAs. Propofol upregulates MIR-143 in osteosarcoma cells, decreasing cancer cell proliferation by MMP13 inhibition via the PI3K-AKT pathway. In gastric cancer cells, propofol exposure induces apoptosis via the PI3K-AKT pathway and MMP2 by MIR-218 upregulation. On the other hand, propofol causes apoptosis by MIR-451 in glioblastoma cells. And in hepatocellular carcinoma, propofol decreases MMP9 expression by PI3K-AKT pathway suppression via MIR-199A upregulation. Anesthetics affect anti-cancer immunity and cell-to-cell communication via microRNAs. Anesthetic exposure during cancer surgery can affect normal cells, anti-cancer immune cells, and cancer cells directly or indirectly via microRNA expression changes. Mature microRNA induced by anesthetics inhibits its target protein expression in the affected cell and in adjacent cells connected by gap junctions. Also, microRNA with RISC is released as EV from cells by exocytosis or in microvesicles, which enables microRNA to alter the target protein expressions in distant cells, including normal cells, anti-cancer immune cells, and other cancer cells. This is what is known as cell-to-cell communication. Next, we looked at two additional topics. The first is that the effects of inhalational anesthetics on cancer cells depend on the type of cancer cell and on onco or anti-cancer effects. And number two, that the mechanisms of inhalational anesthetic effects differ between Sevoflurane and Desflurane. The present study undertook genetic analysis to examine the mechanism of action of inhalational anesthetics using the H4 glioma cell line and the SKOV3 serous ovarian cancer cell line. This slide shows the results of wound healing assay migration test on the left and Ki67 immunofluorescence proliferation analyses on the right. In the wound healing assay, Sevoflurane and Desflurane exposure both suppressed cell migration at 24 and 48 hours after gas exposure. Both inhalational anesthetics reduced the percentage of Ki67 positive cells at 24 hours after gas exposure. It's clear that the inhalational anesthetics Sevoflurane and Desflurane inhibit H4 gliomal cell migration and proliferation. MIR-138 and 210 are known as regulators of HIF-1-alpha, and MIR-335 is reported to control MMP9 expression. To determine changes in their expression induced by Sevoflurane or Desflurane in H4 cells, QRT-PCR was performed just after gas exposure. We found that MIR-138 and MIR-335 expressions were upregulated by Desflurane exposure, whereas expression of MIR-210 was upregulated by Sevoflurane exposure. HIF-1-alpha is a key transcriptional factor in cancers that can be regulated by MIR-138 and 210. Therefore, we examined HIF-1-alpha expression after microRNA inhibition and anesthetic exposure. Immunofluorescence staining showed that HIF-1-alpha was significantly decreased after Sevoflurane and Desflurane exposure compared with the control. Inhibition of MIR-138 and 210 enhanced HIF-1-alpha expression. MMP9 plays a main role in cancer cell migration and is regulated by MIR-335. MMP9 expression was examined after microRNA inhibition and anesthesia. In immunofluorescence staining, Sevoflurane and Desflurane exposure significantly decreased MMP9 compared with the control. Inhibition of MIR-335 enhanced MMP9 expression in the presence of either anesthetic. In contrast, Sevoflurane and Desflurane promote SKOV3 ovarian cancer cell migration and proliferation. In the wound healing assay, the wound gap closure ratio was significantly higher at 4 hours and at 8 hours after Sevoflurane and Desflurane gas exposure compared with the control group. There was no difference between Sevoflurane and Desflurane. Ki-67 immunofluorescence staining showed significantly increased cell proliferation after Sevoflurane and Desflurane exposure, and there was a significant difference between Sevoflurane and Desflurane. Both Sevoflurane and Desflurane significantly suppressed MIR-138 expression in SKOV3 cells. Sevoflurane exposure significantly decreased MIR-210 expression compared with the other groups. There was a significant difference between Sevoflurane and Desflurane, and MIR-335 expression was unchanged after any anesthetic exposure. HIF-1 expression in cytoplasm was elevated after Sevoflurane and Desflurane exposure, which significantly lessened with MIR-138 and 210 mimic treatment in any anesthetic condition. In summary, inhalational anesthetics attenuate H4 cell malignancy via upregulation of anti-cancer microRNA expressions. Sevoflurane exposure to H4 cells decreases H4 cell proliferation and migration via HIF-1 by microRNA 210 upregulation. Desflurane exposure to H4 cells decreases cell proliferation and migration via HIF-1 and MMP9 by MIR-138 and MIR-335 upregulation, respectively. Inhalational anesthetics enhance SKOV3 cell malignancy via downregulation of anti-cancer microRNA expressions. Sevoflurane and Desflurane exposure to SKOV3 cells increases SKOV3 cell proliferation and migration via HIF-1 by MIR-138 downregulation. Only Sevoflurane decreases MIR-210 expression, leading to an enhancement of SKOV3 cell malignancy via HIF-1. In all of these results, the effects of inhalational anesthetics via microRNAs on cancer cells depend on the anesthetic agent used and on the specific cancer cell type. Thank you very much for listening to this presentation about our research.